Hello, welcome to this informal public talk. My name is John Poole. I am a professor in genetics at the University of Wisconsin. And I would like to share with you some things today about the importance of Africa for Drosophila research. First of all, why am I giving this talk? Well, it turns out that African populations and strains of this well-studied fruit fly, Drosophila melanogaster, have been especially important in my own scientific career ever since I was a graduate student. In fact, the first project that I really picked for myself was to try to get a better understanding of this species history within Africa. And this involved assembling a lot of population samples that we did not have in the lab before. And that contributes to, the experiences that I've had and the gratitude that I feel toward researchers that are based in Africa. Especially back then, a number of them shared fly collections with me out of goodwill. And I've also had the privilege of traveling to several African countries for my research over the years. And so, these experiences have, left me with many wonderful memories of the people, the landscapes, the culture, music, food, and so on of the African continent. And these experiences continue to play an important role in just how I look at the world. And so that's one motivation here. And in particular, I have a strong desire to really, help to engage and integrate the African scientific community into Drosophila research on a global level. So I'll get back to that point at the end of this talk. First, to give a little bit of background about this species, if you're not especially familiar with it, this is a very important model organism worldwide for studying a wide array of molecular processes. That's probably what it's most known for. It's been a key model system in genetics for well over a century now. And, it has a very well-studied genome. We know at least something about most of the genes in its genome. But these same properties that make it a really convenient model system to study in the laboratory in molecular biology also make it an ideal species to study in the field of evolutionary genetics, where we're often trying to understand what does genetic variation look like in natural populations, and what do evolutionary processes like trait adaptation or reproductive isolation look like at the genetic level. So this same species, Drosophila melanogaster, is also a really important study system in my own field, evolutionary genetics. And I would say, though, that more broadly as well, even for non-evolutionary studies, natural variation is playing an increasingly important role in the discoveries that are made in this species. And that's really relevant here because Drosophila melanogaster was originally an African species. And this was understood even several decades ago, even just based on the inference that, well, all of its relatives either live on the African mainland or on offshore islands close to Africa. And so it seemed very likely that Drosophila melanogaster was from Africa too. And when we started to look at genetic variation, that became even clearer. Now, today, Drosophila melanogaster lives basically everywhere that humans do around the world. But this is, in evolutionary terms, a very recent situation. The species having originated somewhere in Africa, uh, from there, it expanded around the world in association with humans. And every time a population kind of, jumped into a new geographic area, it only brought with it a subset of the genetic diversity that was present where it came from. So this is known as a founder event population bottleneck, where you have a greater level of diversity in a population that is closer to the original geographic source of a species. But then each time, it overcomes some geographic barrier and gets into a new environment, those founder individuals only carry the genetic variants that are present in their own genomes, and not the ones that are present among other individuals from their source population. So genetic diversity goes down each time the population expands into a new environment. And that would take a much longer time for that pattern to be erased, for diversity to recover in those new populations. So we still see this effect today in Drosophila melanogaster, just as we see it in our own species as well. Certainly Homo sapiens is originally an African species as well before it became a worldwide species. And so the same population genetic processes are in play in Drosophila melanogaster as in our own species in that regard, qualitatively speaking. So I also want to point out that not only do populations from Africa still have the greatest genetic diversity in flies as for in people, that also many African populations show unique phenotypes and have unique ecological circumstances that are not found among Drosophila melanogaster from any other part of the world. And I'll come back to that in a few minutes here. One part of my own research history has actually involved trying to understand what the origins of this species were within Africa. Now, I mentioned that for a while we've had the general idea that Drosophila melanogaster came from somewhere in Africa. But one of the curious things about our knowledge up till very recently of this species is that no one had ever collected it from a convincingly wilderness environment in Africa. And so it wasn't originally clear like where the species would have come from within this vast continent. And so by the mid-2000s, and this came from some early population genetic studies that I and others were involved in, we at least kind of narrowed it down to somewhere in Eastern or Southern Africa. Those populations seem to have higher genetic diversity than those from other parts of Africa. And then as we get into the early 2010s, so this is for me like my postdoctoral years, we started being able to sequence full genomes from a fairly large number of fly strains. And from these full genomes, we could get a better sense of, where within Africa we observe the maximal levels of genomic diversity. And that turned out to be in Southern and Central Africa, so places like Zambia and Zimbabwe. And so this led me to propose the hypothesis that Drosophila melanogaster may have originally evolved in natural environments in this part of Africa, such as the seasonally dry woodlands known as Miombo and Mopane. And so that's a hypothesis, but at that point, we didn't really know, if it did still exist in the wilderness at all, or we didn't, have any idea about how it might have originally become associated with humans, as it is in most parts of the world today. And so one breakthrough came when a colleague of mine followed up on this suggestion and actually found that indeed Drosophila melanogaster persists in the wilderness in Zambia and Zimbabwe, such as in the middle of large national parks far from any human settlement. And in particular, my collaborator, Marcus Stensmeyer, who is an olfactory neurobiologist at Lund University in Sweden, he was very interested in the evolutionary context and the ecological context of the olfactory system that he studied. And he especially wanted to understand, what this fly evolved to be attracted to in terms of smell. And so he wanted to understand, which fruit or fruits in nature might have had particularly important roles in the evolutionary history of Drosophila melanogaster, even before it became human associated. And so Marcus went out there and he found these flies in the wilderness. And before long, other researchers like Daniel Matute and Brandon Cooper discovered melanogaster in other wilderness areas in the same region of Africa. And so it became clear that, yes, indeed, Drosophila melanogaster continues to occupy wilderness areas in this part of Africa, even though it has not been found in the wilderness in other parts of Africa across decades of looking. And a particular key observation that Marcus made was that Drosophila melanogaster was far more abundant when he collected around marula trees in their fruiting season. So marula is, a good-sized tree that makes a fruit with a sweet flesh and also a sizable nut in the middle of the fruit containing its seed. And so, this represents a pretty ideal resource for an organism like Drosophila melanogaster, which feeds on the yeasts that grow on rotting fruit. And so, Marcus discovered that flies were not only abundant around marula. His lab, led by then postdoc Suzanne Mansourian, actually discovered the key molecules on both sides of this attraction, both the compound in marula, primarily responsible for luring the flies in, and the specific protein in Drosophila, this olfactory receptor protein expressed in the antenna, that senses this compound in particular. So a really neat story in terms of, spanning from novel ecological inferences to the key molecular details that underlie that ecology. So now we know where Drosophila melanogaster still lives in the wild, and we even know something about how it lives there in terms of its relationship with marula. And this is all, a pretty new development just in the last 5 years or so. And so marula also has some potentially interesting implications in terms of the evolution of Drosophila melanogaster toward becoming a human commensal species. Outside of these wilderness populations that have been discovered in southern Central Africa, this species is very much associated with humans. And so how did that come to be? Well, marula kind of presents an interesting scenario there. We know that the San people who were who have lived in this part of Africa for a very long time, longer than agriculturalist or pastoralist groups by far. Um, they were um, known to gather very large numbers of marula fruits. And in regions which had kind of natural rock overhangs or caves, they would, gather it into these centralized locations. We know this from archaeological evidence, in part. And so, the fact that humans were assembling these large numbers of fruits that were, fermenting, and I mean, not only that, the San intentionally fermented the fruit to make it alcoholic, and they also stored the nuts for later consumption. So this represents potentially a very abundant and predictable resource for flies if they are willing and able to exploit it. And so in that context, it is notable that compared with its closest relatives, uh, Drosophila melanogaster is much more willing to enter dark places, whether we're talking about caves or buildings. Actually, Marcus Stensmeyer did some experiments in the field uh, in Africa, showing that Drosophila melanogaster was happy to go into one of these caves, in pursuit of a bucket of marula, whereas its close relative Drosophila simulans is less inclined to do so. And another thing about Drosophila melanogaster is that it is more tolerant of alcohol than closely related species. And so this all kind of dovetails into a scenario in which, sometime after the San began to heavily utilize marula and to, concentrate it into their living spaces, there may have been an incentive there for Drosophila melanogaster to become associated with the San populations. And curiously, the estimated timing of when the San used marula to the greatest degree, it actually aligns very well with when my lab and others have estimated that Drosophila melanogaster began to spread around the world. And so this is an example of a study from my lab that actually included a population from a wilderness area that's labeled as Kafu here down at the bottom, which is a national park in Zambia. All of the other populations on this graphic here are human-associated populations. They were collected from towns. Generally, that's where people have collected Drosophila melanogaster in Africa because that's where you could find them. Before we discovered these wilderness populations. And so this is an example of doing something in population genetics that we refer to as demographic inference. So this is trying to estimate important parameters of population history. Things like population sizes, which are reflected by the width, or rather the height here of bars. Each little branch here is one population, either an extant population or the ancestor of two or more extant populations, or the time at which populations diverged. So that's reflected by the number of thousands of years ago on the x-axis here, that a given pair of lineages find their common ancestor. So over on the right-hand side here, this is the present, and then we're kind of going back to the common ancestor of all populations over here, roughly 30,000 years ago, I'm sorry. Flies actually have roughly, we think, about 15 generations a year. And so what we inferred about the history of the human-associated populations is that they began to radiate from southern Central Africa about 13,000 years ago. And again, that is actually exactly when archaeologists estimate that the San's usage of marula was at its peak in this region. Perhaps that was sort of a stepping stone for the species to become human associated. Once it was, perhaps there were other groups of people who were also doing things that were conducive to the persistence of Drosophila melanogaster around their settlements. And so the species expanded, both within sub-Saharan Africa and ultimately beyond. Before terribly long, about 11,000 years ago, we estimate that Melanogaster crossed what is now the Sahara Desert. It was not quite so imposing at that time. It was more perhaps dry scrub and a more limited amount of desert. And at that time, it's thought that people living in the Middle East were beginning to experiment with some kind of pre-agricultural activities, including fig tree cultivation. And so those practices may have provided kind of a soft landing spot for Drosophila melanogaster in the Middle East. Much more recently, we estimate that Drosophila melanogaster managed to occupy relatively colder environments, both within and beyond Africa. So if you look at the blue circles here, these are actually fly populations from relatively cooler locations, either higher altitude locations, like Ethiopia here, and I'll say more about those interesting flies in a bit, or higher latitude places like getting further into Europe. Those events we estimate only happened about 1 or 2,000 years ago. And then even more recently, the species has expanded beyond that into places like Northern Europe, the Americas, Australia, and so on. That's all actually within about the last 200 or so years that the species has occupied, those more profoundly seasonally cold environments, and made its way around the world with the help of human shipping. So, that's kind of a rough history of, what we have learned about the history of Drosophila melanogaster, its African roots and the chronology with which it has spread to, different parts of the world, both within Africa and beyond. So, I want to emphasize, a few particular opportunities that African populations of Drosophila melanogaster, which I sometimes abbreviate as DMEL here. One is that we have this, this newfound, opportunity to study wilderness populations of this species, and to understand how perhaps the species adapted to human-associated environments. So what are the differences between wilderness and human-associated Drosophila melanogaster populations? It is also a very rich source of diversity for studying the genetic basis of adaptive trait evolution. Clearly, Africa itself contains such diverse environments, and contrasting climates, and the species has needed to adapt to these as it has spread within Africa. And so that provides abundant raw material for adaptive trait evolution, which my lab and others study. And it also provides some nice systems for studying some of the very earliest steps toward what could ultimately become speciation in the absence of sufficient gene flow. And I'll say more about each of these things in turn, starting from this, this idea that we can use African Drosophila melanogaster to study adaptive trait evolution. I've kind of already talked a little bit about the first point, and I'll come back to the last one. But I'd like to emphasize here that, again, this species has expanded into a range of very diverse environments, including just within Africa. And that does provide an incentive for various traits to evolve. And so, one fundamental evolutionary question is, how does that happen at the genetic level? when a trait evolves, is that because of one gene or 10 different genes or 100 genes? And what's going on in terms of genetic variation? Is natural selection favoring newly occurring mutations, or is it mostly favoring standing genetic variation that was already around before the flies went into a given area? And does natural selection actually raise the frequencies of those favored variants all the way to 100%? Or does it not always need to? Is there sometimes more variation than is needed, really, to get this trait to its new optimum or threshold value? So these are some of the questions that we're interested in. And I'd like to particularly point to this unique population, that I was fortunate to encounter from the Ethiopian highlands. And here it turns out that the Drosophila melanogaster found at these extreme altitudes in Ethiopia where people live, which can be 3,000 meters or more above sea level. So these flies are the largest individuals that we find in any natural population. They are much darker than any other known melanogaster, which may be an adaptation to provide protection against increased ultraviolet radiation at those altitudes, at least in part. And another striking thing about these flies is that they lay many fewer eggs than populations from lower altitude environments. It seems like perhaps they are focusing a little bit more on survival as opposed to reproduction. Although, this might not seem like the harshest environment in the entire world, um, there's a lot of pressures that they need to adapt to, and in particular, this is an area where it's persistently somewhat cool all year round. And so, whereas where I live in Wisconsin, the species is here, and although it needs to hide out in the winter, it can always wait for summer, and then summer is pretty good for these flies here. But in the Ethiopian highlands, they can't just wait for summer. That's not happening. It's similarly cool year round. And so they just have to be able to develop in a suboptimal temperature. And they have to deal with, the reduced air pressure and the issues of oxygen and desiccation that go along with that, in addition to other things like the UV I mentioned. So in terms of the phenotypic readouts, it seems like this was a particular challenge for the species. And so it seems like perhaps they're investing more in survival and less in reproduction. And I also see signs that they may be provisioning each offspring to a greater degree. And they do actually produce pretty robust individuals as well. They're not just large, but they show particularly strong just survivability and resistance to specific environmental challenges, especially desiccation, which makes sense given that, thin, high altitude air is a very desiccating environment. So this is an example of one particular population that provides considerable raw material for studies of adaptive trait evolution. And so what can we do to learn about how adaptive evolution works at the genetic level in this genetic model organism? Well, one thing we can start with is doing mapping experiments where we cross strains that have different traits because they come from different geographic populations. And we can perform what's called quantitative trait locus mapping, or QTL mapping. This is an example of that using bulks of offspring that have contrasting phenotypes and subjecting them to genomic sequencing. So this is an example from my lab where we were mapping a particular pigmentation trait in these Ethiopian flies, crossing them to flies from the ancestral range, in this case from Zambia, lighter bodied flies. And so here each peak that is labeled with a Q is statistically significant and indicates that there is a gene in this part of the genome that contributes to the trait difference between the parental strains that were used. So, that gets us to one block of the genome. And then, within each little QTL peak, as we call them, one approach that we could take is to look for genes that have particularly strong allele frequency differences between the source populations. So here is a well-known pigmentation gene that happens to reside within I think the tallest peak on that last mapping plot. This is a gene called ebony. And so we've known for a long time that when ebony is mutated, you get darker flies. And so here we can see that when we're plotting a statistic that summarizes allele frequency differences between populations, this FST statistic, we can see that at the ebony gene region, and in particular, in this intronic region of ebony, there are certain genetic variants, certain single nucleotide polymorphisms with very strong allele frequency differences between Ethiopia and Zambia. So variants that might, let's say, be at 3% frequency in Zambia, but 80 or 90% frequency in Ethiopia. And so it is possible that variants like this may have been acted upon by locally adaptive natural selection. And so this provides a good foundation for more detailed studies at the molecular level, leveraging the rich toolkits that we have available in this model system. Even things like CRISPR for making transgenic flies, and these things can help us to confirm, potentially even which were the specific mutations responsible for evolutionary change. But of course, this is ultimately, all dependent on, having the right raw material to study, which in this case was the biological diversity of African Drosophila melanogaster. Of course, it's not only Drosophila melanogaster that allows us to make interesting evolutionary inferences. The knowledge that we have about Drosophila melanogaster has actually allowed the entire genus to become, kind of like a model system in evolutionary genetic research. This is a very broad genus. And there are thousands of species that exist, which fall within Drosophila. And I would suggest that probably hundreds of species of Drosophila live in Africa. Some we know about, and I am certain that there are many we don't know about yet that just haven't been described from wilderness African environments. So these species encompass a vast array of biological diversity and many different ecological lifestyles, all of which can provide intriguing raw material for biological study with the aid of the knowledge of the Drosophila melanogaster genome, and in some cases tools that can be exported to other species as well. And so, in particular, the close relatives of Drosophila melanogaster, which I mentioned are from Africa or nearby islands, have been particularly fruitful targets for evolutionary genetic studies of many different kinds. This is just highlighting a couple of different systems. So Drosophila seychellii at the very bottom here is pictured here on its favorite fruit. This species is from the Seychelles, and it lives on this fruit called morinda or noni, which is otherwise toxic to Drosophila, but this species has evolved to tolerate these toxins because this fruit is the most dependable resource available where it lives. Above that, I'm showing a composite image of two closely related species, Drosophila yacuba, which exists across most of sub-Saharan Africa, and Drosophila santomea, which is an island endemic native um, to Sao Tome and to another nearby island as well. And this comparison has also been of interest in terms of the um, ability to study the genetic basis of traits such as pigmentation that differ between these species. And I would also point out that a former postdoc in my lab, Amir Yassin, actually discovered a special island population of Drosophila yacuba, which had recapitulated the evolution of Drosophila seychellia. It had become a noni specialist as well on the island of Mayotte, between the African mainland and Madagascar. So there's plenty of interesting biology to be observed in other Drosophila species as well. And so, this has also been a major target of evolutionary genetic study, especially in these closest relatives of Drosophila melanogaster, but also in other species as well. And so one example that I would point to here is that the relatively closely related species, especially the closest relatives of Drosophila melanogaster, which are Drosophila simulans, which is another kind of widespread species that lives across a good bit of the world, and it's also human associated like melanogaster, although it is thought that simulans originated from Madagascar. Simulans, though, has a couple even closer relatives called Drosophila mauritiana from Mauritius, of course, and Drosophila seychellia, as I mentioned, from the Seychelles. And so studying the genetic basis of reproductive isolation, both within this simulans clade here and between those species and melanogaster, has been the source of some key insights in terms of the genetic basis of species isolation. I would also point out, though, that much earlier stages of reproductive isolation are also visible even within Drosophila melanogaster, even though these human-associated populations only diverge from each other within the last 13,000 years, as I mentioned earlier. One thing that we know to vary is mating preferences. So, there's kind of, I would say, three broad realms of mating behavior that we know about within Drosophila melanogaster. There is southern and eastern Africa, and there's a unidirectional mating preference between that realm and flies from elsewhere. And there's also kind of a Western and Central African realm of mating behavior, and a separate unidirectional mating preference that separates those populations from other melanogaster. And so, this provides additional raw material to understand how is it that natural populations begin to become reproductively isolated from each other. And another example is that actually, if you do crosses between fly populations from certain populations, like if you cross some flies from Zambia to flies from Europe, the first generation offspring look fine. But when you get to the second generation, a subset of those F2s actually seem to suffer some fitness defects. And this is known as hybrid breakdown. And my lab has observed this particularly in the context of male reproduction. A subset of the F2 males are completely unable to reproduce, and others may be impacted to more moderate degrees. And so, these systems within melanogaster provide really unique opportunities to ask, what is going on at the very earliest stages of reproductive isolation? These populations can absolutely still, exchange genes between them, and they do. But at the same time, there are barriers. There are reproductive barriers to some degree, both in terms of pre-mating barriers and also in terms of, post-mating barriers, even at the, later generations of admixture here. And so, these give us kind of a tantalizing opportunity to study, early stage reproductive isolation within a genetically tractable model system. So, I think although, we continue to make progress on these questions, there are still some very tantalizing scientific opportunities available from African Drosophila melanogaster. One that I alluded to before is, trying to understand the differences between wilderness and human commensal populations of melanogaster. And so, I'm interested both in, to what degree do any visible traits differ between them, but perhaps especially, to what degree does their behavior differ? And what are the genetic underpinnings of that? And with regard to the wilderness populations, we'd like to know more about how they are structured in nature as well, and how do they differ in their population genetics from human-associated populations. And getting back to what I was just talking about, we also don't really know at this point to what degree there is reproductive isolation between wilderness and human commensal populations. Can they tell, are there mating preferences that differ between them? And are there any, post-mating consequences if they do interbreed? We don't know. And so that's one key area of opportunity, I think, based on the recent discovery of the wilderness populations. Another opportunity that hasn't been investigated thus far is to try to study seasonal evolution in African melanogaster populations. In temperate parts of the world, especially the eastern US, and also Europe, there has begun to be, very large-scale studies of seasonal evolution at the genetic level in this species. I mentioned that melanogaster probably has roughly 15 generations per year. And, in places like the US, they're actually in profoundly different environments throughout a summer generation compared to, throughout a winter generation. And so the selective pressures between seasons are actually very different. And, researchers have discovered that there are, a decent number of genetic variants out there that seem to change frequency predictably from one season to the next. And likewise, there are certain traits that change predictably from one season to the next at the genetic level, even when you take them into the lab and raise their offspring in a constant environment. And so we know that this happens in temperate regions. And we don't know to what extent this happens in tropical regions, where often the primary seasonal variables are, wet season versus dry season. And we also don't know to what degree, even the same polymorphisms, at the genetic level could potentially underlie seasonal evolution in both the ancestral range of the species and in more derived areas. To what degree are there commonalities between the ways that flies adapt to winter and the ways that flies adapt to the dry season, for example? Perhaps neither one is a good time to put a lot of energy into reproduction, and a much better time to focus on survival. So this is another clear area of opportunity. Another kind of lingering mystery is, why do we see chromosomal inversion polymorphisms segregating at fairly appreciable frequencies in many populations, especially those in Africa? So an inversion, of course, is just a piece of the chromosome that is flipped around. And in Drosophila melanogaster, it's been known for decades that there are multiple inversions that vary within natural populations. Uh, and in spite of, some geographic associations that different inversions have, in general, we don't know a lot yet about why they segregate at the frequencies that they do. They tend to be at the highest frequencies, actually, in populations from relatively, milder, more permissive climates. Not all of the inversions, but many of them show that pattern. But what are they doing there? These are environments relatively similar to that which the popula to that which the species originally evolved in terms of climate. And these are all derived inversions, so it's not clear that they would have been necessary to adapt to these ancestral environments, but they may modulate important trade-offs that are subject to locally varying selective pressures. But we're still really trying to get a handle on what trade-offs those are that these inversions are modulating and why they're often held at a given frequency in a given natural population. So the last part of my talk today, I'd like to focus on, the desirability of really integrating African science into the study of African Drosophila. Um, that may seem like a no-brainer, an obvious thing, but unfortunately, it is not really the case today. Thus far, African Drosophila, both melanogaster and other species, have largely been studied by non-African researchers. Especially from the United States and Europe. So people who look like me, basically. And that is far from ideal. And it would be a great thing both for global science and I think for African science as well, if there was far more integration of African researchers into this enterprise. Why has that not been the case? Well, I mean, funding is often a challenge. They're and relatedly, there can sometimes be perhaps a disconnect in terms of the focus of science. So I understand this from having traveled in Africa, that often, if I told people that I was there to study Drosophila melanogaster, I would get some very surprised reactions. This tiny little fruit fly of no real economic significance or direct social impact. Most studies on Drosophila, and certainly, most evolutionary studies in Drosophila are very much basic science. So we're trying to learn about fundamental biological processes. Obviously, you need a strong basic science foundation if you are going to make more applied discoveries down the line. But understandably, in my opinion, most science that is going on and most science that is funded within Africa is targeted toward urgent social needs. So, health problems that are affecting people in this country right now, limitations on the production of food, and perhaps to some degree also studies focused on species of local conservation interest. And so it's not hard to understand why that is the case, given somewhat limited resources in most countries in Africa. And yet, I would argue that every country does need basic science, if only to to provide the foundations of training its future generations of scientists. And this is not to say that basic science does not exist in Africa. Clearly, it does. But I think, at least in my experience, this has been kind of a barrier toward being able to successfully engage the interest of African collaborators. Uh, and so what do we need? I wouldn't say that I have, the magic set of answers, but, my general sense is that, I mean, what we need for this integration is obviously collaborations between the people who are studying African Drosophila now and people who are in Africa and would like to join this effort. But to make that happen realistically, I think it's going to require collaborative grant-seeking exercises. There needs to be some way, I think, of pursuing funds either from places like the U.S. that have relatively more scientific funding, or from international agencies in some cases, and for at least a portion of that funding to go to collaborators in Africa. So we need to figure out how to make that happen one way or another. If it was easy, it probably would have already happened. Nevertheless, that's something that clearly needs to happen. And another thing that I think would be really important is to provide more chances for students and postdocs from Africa to pursue at least a part of their training abroad, somewhere like the United States or Europe, for example. And so, I work, with the genetics PhD program at the University of Wisconsin, including with our admissions committee. And I was so glad, recently when we were able to accept our first student from Africa, an applicant from Nigeria. And so this is something that I really hope to see more of in the future, not just in our program, but across the board, because, it is clear that, that studying, that training students who come from, countries that have a lot of native biodiversity, is a very high impact activity for any graduate program to take on. So that's something that I hope can happen more and more in the future. I would also like to point out one existing initiative that is already trying to encourage more researchers in Africa to study Drosophila. Now, this is called Dros Africa, and Dros Africa does have a focus that is kind of distinct from what I do. I'm an evolutionary geneticist. Dros Africa has a more biomedical focus, in particular, the idea of using Drosophila as a very cost-effective system to screen drug candidates that might contribute toward treating human diseases. And that's a very exciting direction as well. And Dros Africa has already hosted multiple workshops in different African countries. So, this is a great model for, the sort of, interaction and collaboration that I hope can happen across a broader realm of Drosophila research. So to summarize, my main messages in this talk, clearly Drosophila is an important model organism, in a lot of different areas of biology, including my own field of evolutionary and population genetics. I've tried to highlight that this is originally an African species, and that before it became a human commensal, Drosophila melanogaster was perhaps a marula fly, living in the woodlands of southern Central Africa. And then, after it became human associated, we estimate that the species expanded both within and beyond Africa within the past roughly 13,000 years. But even today, African populations still have the most genetic diversity of any worldwide populations of Drosophila melanogaster. And they have some really exceptional phenotypic diversity and some interesting initial stages of reproductive isolation as well, all of which continue to make these African melanogaster populations very important in basic evolutionary genetics research. But this research would benefit from a greater involvement of African scientists. And I would even say that in the long run, they are the ones who should be leading most study of African Drosophila. So again, I don't have all the answers for how to make that happen, but I'm glad to serve as a point of contact, and feel free to get in touch with me if you would like to. I hope that you've enjoyed this presentation.